Good afternoon, everybody. Thanks to attend this concurrent session on the molecular aspect of plant-fungal interactions. So we had two sessions which split because there were too many potential speakers and posters you will see in the poster session. So we decided to have two sessions, one dedicated on effectors today, and on Thursday, one more dedicated to the mechanism of infection. So I invite you also to come on Thursday afternoon to the concurrent session that will be headed by Antonio Di Pietro and Nathalie Poussereau. So here I am Marc-Henri Lebrun, and it's not Ely OLIVEIRA-GARCIA, but Andrea Sanchez. So Ely could not come at the last minute. At the first time, we were thinking also Barbara Vallon could come, but finally, Nick Talbot will replace Barbara and Ely to speak about their joint project toward understanding how Magnaporthe oryzae co-opt plant endocytosis for translocation of cytoplasmic effectors, how they get into the plant cell. Nick?
Thank you. So, thanks very much. I'm pleased to be here. But I really feel sorry that Ely couldn't come, because it would have been really great for him to have been able to present this work, which he led. But unfortunately, he had a visa issue which prevented him from attending the meeting. Okay, how do I start? Does it come up automatically, or do I have to click? Okay, great. Okay, so the work that I'm going to talk about was a collaboration really between Barbara Vallent's lab. Ely started as a postdoc in Barbara's lab and then had his own group at Louisiana State University and our group at the Sainsbury Laboratory. And what we were interested in doing is trying to understand how Magnaporthe or effectors are taken up by plant cells during infection. So what do we know about effector secretion by the fungus and uptake? Well, at the start of this project, what we knew is that effectors in Magnaporthe really can be divided into two main groups. There are those that are destined for the apoplast, which is the gap between the fungal cell wall and the plant cytoplasm. So those apoplastic effectors, and those are secreted through conventional Golgi-dependent secretion at the hyphal tip during invasive growth. But there's a second group of effectors, and those are a well-known group of effectors, which include most of those that are recognized by intracellular immune receptors, the AVR effectors. And those are, of course, destined for delivery into the plant cytoplasm or indeed to organelles within rice cells. And those are delivered in a different way. They go through an unconventional secretory process, which is Golgi independent, but still exocyst dependent. And therefore is Bruffeldinase insensitive. And they appear to accumulate in a structure called the biotrophic interfacial complex, which was discovered in Barbara Vallent's lab. And we noticed for many years that effectors will accumulate at that site. And a number of independent lines of evidence, including photobleaching experiments, have suggested that the BIC is a potential portal for delivery of effectors into the cytoplasm. But the mechanism remained unknown. So we formulated really a hypothesis, which was to test whether cytoplasmic effectors are delivered from the BIC and internalized into the rice cytoplasm using endocytosis. And we set out to really investigate whether that was the case. So this is what the BIC looks like under laser confocal imaging. And what you can see, especially under super resolution levels of resolution, is that the BIC, this bright punctate structure, initially at the tip of the penetration peg, but then ultimately on the side of the invasive hypha as it differentiates. If you look under very high resolution, you can see that there are actually puncta within it, and we call those membranous effector compartments. So those are labeled with an effector. This is the PWL2 effector, which you'll hear more about at this meeting. PWL2 is an effector which also acts as a host specificity determinant, but is often used as a marker for cytoplasmic effectors. And in contrast, apoplastic effectors will accumulate in the periphery around invasive hyphae, although BAS4, which binds to membranes too, actually still has some localization to the BIC. But what we see is a big group of membranous effector compartments, these membrane-bound vesicle-type structures within the BIC. And that was the first observation. These multiple vesicle-like structures which contain cytoplasmic effectors. And what we found is that there seems to be some dynamism to these structures. The mechs within BIC structures, the BIC that forms at the penetration peg tip, the so-called tip BIC, actually has smaller, more dynamically moving MECs, and then those increase in size as the BIC matures and the invasive hyphae start to ramify through that initial colonized cell. And we can also see a difference in both the number of BICs, which is greater in the initial BICs, so suggesting there may be some membrane fusion going on, vesicle fusion, and also they're larger in the mature BICs, which again suggests that there's some dynamism in the way in which those are being traversed. So how far do the BICs move? Well, we can begin to see them in the cytoplasm around the BIC, but they're seldom more than about 7 micrometers away from the BIC itself. So they normally are in very close proximity to the BIC, and then they disappear. And in fact, you can see them bursting and releasing their contents. So they appear to be very closely associated with the BIC, but you see some examples of them rarely in the cytoplasm. And you can see that both with PWL2, and in this instance, PWL2 is being labeled with a nuclear localization signal to target it to the nucleus so that we can see it. So it just increases its relative concentration in the cell. But BAS170, which is actually a nuclear localized effector, actually goes naturally to the nucleus. That one too, so you can see it lighting up the rice nuclei in this panel. But you can also see that there are small vesicles containing BAS170, and another cytoplasmic effector that accumulate and are in close proximity to the BIC. So they're rarely observed at a long distance away from where the BIC is observed. So what we decided to do is to try and test the hypothesis that these MECs are associated with endocytic uptake into rice cells. Now, of course, endocytosis comes in 2 flavors: clathrin-mediated endocytosis and clathrin-independent endocytosis. And with clathrin-mediated endocytosis, we see both clathrin heavy and light chain forming associated with the adaptor protein complex and coating these pits, which eventually will bud off as endosomes. And those are very often associated with actin. So in fungi and in mammals, actin is absolutely essential for clathrin-mediated endocytosis. In plants, the system is slightly more complex, but there's an actin close association, particularly with endosomes and endosomal movement. So here are the questions, really. Does clathrin-mediated endocytosis facilitate the internalization of those effectors? And can we see any association with actin transport within the rice cytoplasm? Well, first of all, we notice that there's a close association with the plant plasma membrane. This is a rice transgenic line which we made, which has GFP targeted to the plasma membrane. And you can see that the BIC itself is composed of plant plasma membrane. And indeed, the MECs themselves are bound by plasma membrane, which is of plant origin. So that's consistent, again, with an endocytic origin of them. We also find another effector, which is really quite intriguing, because this is an effector which is present in membrane-bound compartments, but they seem to be a distinct subpopulation compared to the MECs that carry the bulk of the cytoplasmic effectors. And if we look in more detail about the localization of BAS83, then it appears to be localizing to the extracellular invasive hyphal membrane, the plant membrane that invaginates and surrounds the invasive hyphae. And the hypothesis which is being worked on in Ely's lab is that BAS83 may be involved in membrane biogenesis. It may be that effector-driven membrane biogenesis in the plant is key to infection, key to the accommodation pathway that's allowing these fungal invasive hyphae to grow and in such a large volume within an epidermal cell, and he's actively testing that hypothesis. Also, if we take a rice transgenic line expressing actin, then we can see there's a lot of actin association with the BIC. So this is a life actor GFP line of rice, and you can see that there's a strong association of actin with the BIC, and you can also see that using phalloidin staining too. So there is a strong actin association with the BIC, and we can actually see it tethered to the rest of the actin cytoskeleton under high resolution. If we then localize clathrin light chain, GFP, then we can begin to see that, that there is an association of clathrin with BIC structures too, and indeed with MECs. So we see actually a very strong co-localization between any cytoplasmic effector that we've looked at when we've tagged that to a fluorescence marker, and with this, in this rice line expressing clathrin light chain. But interestingly, some of those clathrin light chain vesicles don't contain effectors, which suggests, of course, that they may have other cargo. So there's bound to be other cargo that are being taken up by plant cells in addition to effectors. But we can certainly see an association with every time we look at an effector with these CLC vesicles. And they again appear to be dynamic. So here's the BIC structure, and you can see composed of these membranous effector compartments containing, in this instance, PWL2. And you can see that there are other sites of endocytosis that one can see across rice cells, but the bulk of the strong fluorescent signal associated with the BIC as the invasive hypha enters the rice epidermal cell. So how do we test that then functionally? Well, what we used was virus-induced gene silencing VIGs. This is using the Brome mosaic virus system. And we silenced two components of the clathrin-mediated endocytosis process pathway, the adaptor protein complex 2 gene, AP2, and also clathrin heavy chain. Those VIGS lines are really reduced and stunted in growth, as you might expect for something which is such a crucial component of the endocytic process in plants. And so you can see that those plants tend to be stunted. At the same time, there's, although Ellie hasn't shown it in these slides, we did a control with clathrin-independent endocytosis where we silenced flotillin. So flotillin is a key component of clathrin-independent endocytosis. Which of course normally involves sterol-rich lipid raft type membrane compartments too. So we also silence that at the same time, which also caused some stunting to occur. But what we found is that when we actually inoculated those VIGS lines with rice blast, then we saw resistance in the clathrin heavy chain mutant and in the AP2 mutant. So there was a significant reduction in rice blast symptoms. And indeed, we still saw symptoms in the flotillin line, which again isn't shown here. But the flotillin line actually still showed symptoms, suggesting that this is something that's specific to clathrin-mediated endocytosis. When we actually look at the MEC formation, the formation of these membranous effector compartments, then that's significantly reduced in compared to the control. So here's the control. You can see lots of those MECs around the BIC, but you can see now we get swollen BIC structures, and very few of those membranous effector compartments are visible within around the BIC structure. So we get swollen irregular BICs, and it seems to block formation of those vesicle structures. So in parallel, we carried out a whole series of chemical inhibition experiments using a variety of different inhibitors of clathrin-mediated endocytosis, and indeed clathrin-independent endocytosis too. And you can see using triclosan, cantharidin, chlorpromazine, wartmanin, but also in particular the specific CME inhibitors, ES9 and ES917, which inhibit clathrin heavy chain. That in those cases, again, we could impair the ability of those MECs to form. So we saw far less of the membranous effector compartments. And instead, we saw swollen BIC structures. And those BIC structures even appeared under the aprosorium. So very early during plant infection, we started to see an accumulation of cytoplasmic effectors in a very large compartment, but we didn't see those small membranous effector compartments forming in the presence of those inhibitors. And in contrast with the clathrin-independent endocytosis inhibitors, which we looked at, methyl beta cyclodextrin, for instance, or philippin, we didn't see any similar effect. And in contrast, if we look at apoplastic effectors, so again, here we're looking at BAS4, then BAS4 will travel normally to the interface between the fungal cell wall and the rice plasma membrane. It still accumulates at higher concentrations at the BIC because of this membrane affinity. And that's in contrast to the fact that PWL2 now no longer forms MECs. In this instance, sorry, compared to, I'm showing you the control here, with the, where it does form MECs compared to here where there's no MEC formation in PWL2, but the BAS4 localization is more or less the same as in the control. So that really suggests that cytoplasmic effectors are being affected by impairment of clathrin-mediated endocytosis as a consequence, and we've either genetically or using inhibitor studies. So just to summarize the work and where we are currently, the working hypothesis, which is being now actively tested really in Ellie's lab and in Barbara's lab, is that the cytoplasmic group of effectors, including the major AVR effectors that are recognized within the cytoplasm or have targets within the cytoplasm or in organelles within rice cells, appear to be taken up by clathrin-mediated endocytosis into these membranous effector compartments, which then will lyse and release the effectors directly into the cytoplasm. And this is associated with this large membranous structure, the BIC, which is composed of rice plasma membrane, and also plant actin, which accumulates to very high concentrations at that point and seems to tether that structure within the cell. And that's in contrast to the apoplastic effectors, which of course are going to carry out suppression of extracellular immunity, like chitin-triggered immunity, for instance, which are being conventionally secreted and will accumulate in this in the apoplast, and those appear to be unaffected by impairment of any of these processes. And he's now testing that process and also testing the role of BAS83, which may be an effector that's associated with the stimulation of membrane biogenesis. And in my group, we're now really investigating the sorting mechanism that enables effectors to be sorted into the apoplastic or the cytoplasmic route for fungal secretion. So we're focusing much more on the fungal side of the interaction and trying to work out how that sorting mechanism works, whereas Ellie is actually looking at the uptake directly into the cells in collaboration with Barbara's group. So the summary really is that we've uncovered some evidence that clathrin-mediated endocytosis has an effect and is important for effector uptake by Magnaporthe oryzae, and you'll hear later in the session some completely independent but parallel observations in the oomycete pathogen from Paul BIRCH and his group. So he'll tell you about that afterwards. So just to acknowledge the work, so Ellie really led all of this work and carried out the overwhelming majority of it. He was assisted by others in Barbara's group. The VIGS lines were made in Rick Nelson's group at the Samuel Roberts Noble Foundation. In my group, the key people were Clara Rodriguez-Herrero and also Magdalena Oderoth, who did the work. And this work was carried out both in my original lab in the University of Exeter and then latterly at TSL. Ellie's work's funded by the Board of Regents in the state of Louisiana. Barbara's work also by NSF, and my work by the ERC and Gatsby. So thank you.
So maybe first we will start by if you have some few questions from the audience, and then we will use the app. So, Ralph, you have a question?
Yes, thank you, Nick. Great talk. Ellie's doing some fantastic work. But I wanted to follow up on just something that you alluded to at the end, and that is, you know, the thing that intrigues a lot of people is the sorting mechanism. Can you expand on that a little bit?
Well, I can only tell you about a bunch of unpublished observations, really. So Barbara noticed several years ago that that if you did promoter switch experiments, you could actually redirect effectors from the cytoplasmic route to the apoplastic route. And we investigated that in a lot of detail. We actually did promoter and signal peptide switches. And we found, indeed, that's true in our hands, in our lab. We can actually show that you can direct an apoplastic effector to the cytoplasm or a cytoplasmic effector to the apoplast simply by changing the promoter and the signal peptide. Our working hypothesis is that perhaps this is not to do with the signal peptide per se, but maybe the 5' UTR. Maybe this is an mRNA sorting mechanism. So that's the hypothesis we're testing. We don't have any data yet on that, but the hypothesis we're testing is perhaps there is a sorting mechanism at the level of mRNA, and that directs it into the appropriate spatial configuration that it enters that pathway. We may be wrong, we may be completely wrong, but that's what we're testing, because we're really intrigued by why you see that clear ability to switch the destination of an effector during secretion.
Question from the audience, yes?
Hi, Nick.
Hi, Petra.
Great stand-in job there. I was just wondering, do you think there's a specific mechanism for the potential bursting of the mechs? Or do you think it's...
Yeah, that's a very good question. I think we don't know. You know, we observe it, we've seen it happening, we see the bursting happening, but whether that is a controlled, again, effector-driven process, whether that's something that is regulated by the normal plant endocytic mechanism, we don't know. It's going to be intriguing to see whether those MECs actually are acting in the same way as plant endosomes or not. They seem very different. They seem very different in size. They're much larger. I didn't really talk about the size, but these range in size from about 200 nanometers up to around 600 nanometers. So they're much, much larger than you would normally predict. But how they're actually then bursting and whether that is a controlled pathogen-driven process, we don't know. But we'd love to know that, and I'm sure that's something that Ellie in particular is going to be exploring too.
So if there is no more question from the audience, then I have maybe two small questions. So one is, how do you think could effectors been escaping the normal secretory pathway and then get into the beaks? Any idea?
Yeah. No, I don't know. And in fact, you know, I still think we still need to try and find some more direct evidence for that route into the BIC. I can tell you another experiment we're trying to do is to use photoactivated GFP to try and see if we can actually look at the temporal dynamics of entry to the BIC. That's really hard, challenging to do, but that's how we're trying to address that. But we don't know really, I think, how that mechanism works. But the sorting mechanism I discussed there with Ralph, I think, gives us some clues to the way we think it might be happening. But of course, all could be happening at the level of the protein rather than transcripts. So we don't really know.
Okay. And then the last question is, how do you think that this recruiting of the plant endocytosis system could also affect the plant endocytosis itself, which means that maybe that it cannot recruit things during the infection? Or
Yeah, absolutely. I mean, it may well be that this is, you know, all part of the sort of overall susceptibility that you're impairing by not only by sort of hoodwinking this system and hijacking it, that you're also preventing the plant from undergoing other forms of endocytosis, which might be important in immunity. So I guess that could be the case, yeah.
Okay. Thank you very much, Nick Talbot, and thanks for replacing at the last minute the speaker. So the next speaker is Sylvain RAFFAELE from INRAE Tolosan, Castanet Tolosan, France, and he will speak about more structural biology of effectors.
All right. Good afternoon, everyone. So I'd like to thank the organizers for this opportunity to present today and you all for attending. So I'm going to tell you about structural genomics to study the evolution of effectors. So to get started, very broad general primer on what effectors are and the molecular basis of plant pathogen interactions. So pathogens on this side of the diagram secrete molecules that interfere with the functioning of their host. That can be called with the general term of effectors. So in turn, the plant has an immune system that recognizes effectors as well as other molecules produced by the pathogens. And among the responses, you have the well-known cell death hypersensitive response, but also the production of antimicrobial molecules that in turn can be counteracted by detoxifying molecules from the pathogen, so in some kind of a toxin-antitoxin interaction. So in summary, you have several molecular dialogues taking place between these two organisms. And you can imagine that when the host population changes, this whole pattern of molecular dialogue is completely changing too. So this is one of the questions I'm very much interested into, trying to understand how these dialogues evolve after a change in host. So broadly speaking, we can distinguish two major trajectories. In the one hand, you have host switching, which is the situation where closely related species of pathogens are able to infect specifically different plants, but they don't cross-infect. So this is the case, for instance, in the late blight pathogen. You have here different related Phytophthora species that specifically infect different botanical families. And this is because, at least in part, because there are trade-offs in the activity of some effectors on the different hosts. But there are also different situations where the host range is expanding, meaning that you don't get this kind of trade-off. You actually have an increase in the number of species that the pathogen can infect. This is the case, for instance, in the white mold lineage here, the fungus Sclerotinia, with some species being able to infect only a few botanical families, whereas devastating pathogens are able to infect much more diverse range of hosts. So we can imagine that the constraints on these different effectors are quite different, and I thought it would be very interesting to figure out how specific and diverse the effectors can be in this type of lineages. The problem here is there's little information, functional information about those. And so we're a little bit stuck to characterize this diversity. Well, basically because there's about a quarter of these effectors that have absolutely no known domains, so this is very difficult to infer any function from those. So we decided to use structural predictions to do that, thanks to recently developed tools. And so the design we have chosen is depicted here. So we took 20 species of ascomycete fungi with different lifestyles. So some of them are plant pathogens, other are animal pathogens, even mycoparasites. And we also took non-pathogen species in there. And we also decided to focus on what we called the orphan candidate effectors, with absolutely no homologs and no non-functions to really target the dark matter that I mentioned. So incidentally, it turned out that Siong and Krasileva had taken and were already quite well advanced in a very similar approach with slightly different design. So they also analyzed about 20 species, but including basidiomycetes and oomycetes. Also taken plant and non-pathogens, but they analyzed all secreted proteins. So the nice things about these two parallel studies that we could find different, like the same, we had the same findings independently. So these major findings are as follows. So one is that we find structural families of effectors that are very abundant. So in our study, we find that over 65% of effectors that we analyzed at over 2 analogs in terms of structure. And using just 62 families, we can describe about, well, the majority of the effectors from the 20 species we analyzed. So you have here representative structures of these well-conserved families. And another interesting finding was that some of these families are very broadly conserved. We found 18 families that were conserved in over 80% of the species that we analyzed. And specifically, there were 46 that were shared by pathogenic and non-pathogenic species alike. So this is what you can see in these diagrams. Each dot is a structural family of effector, and you can see they sit in the middle because they are present in similar amounts in animal pathogens, in plant pathogen, or fungal pathogens, in the different fungal lineages that we analyzed, and with in fungi with different lifestyles, either non-pathogenic, necrotrophic, or other lifestyles. So these were findings in agreement between our two studies. Next, we wanted to get some insights into why these families have been quite cryptic until recently. And what we found is that they actually have a very well-conserved structure in spite of very divergent sequences. So if you plot here the sequence divergence relative to structural similarity, you see that there are some guys here that diverged a lot in terms of sequence, but still look very similar. This is what you can see in this little movie here. The different proteins you have here have less than 10% identity, pairwise identity, so there's very little identical to all of them, but still we can nicely recognize the same fold. So this is why we cannot delineate these families by classical blast and alignment strategies. But using deep HMM searches, we are able to reconstruct some of these families and actually expand them, sometimes beyond the fungal kingdom. And this finding also suggests that some of these families might have very ancient common ancestors. So overall, this means that we have here ancient folds that remained adaptive in lots of different contexts, in different species with different lifestyles, and this in spite of very high sequence divergence. So the question then was, what makes this structure robust to mutations? To answer that, we performed virtual mutations and predicted structures of these virtual mutants. So we did that in different ways, either mutating amino acids one by one, such as through deletion scans, or mutating amino acids several at the same time. So for instance, here you have shuffling of mutations over 5% of the whole sequence. What was a bit surprising in these scans were that we had lower structural divergence than expected when we mutated several residues compared to when we mutate just one of them. So we could derive from that some kind of a metric that describes the effect of mutation relative to other mutations that could occur in the effectors. So some of them tend to reduce the divergence that is caused by co-occurring mutations. So we called these robust mutations, and others tend to increase the structural divergence, and so these are labile mutations. So then we can spot this metric on the different structures. Well, we see that there's no clear pattern here. Well, the property tends to be a little bit random all around the structure. So if our hypothesis that some mutations allow these structures to stay stable and robust, we're expecting the robust mutations to co-occur with, well, other type of mutations. So to test that, we analyzed extant diversity in these effector families, looking for patches of co-mutated residues. So you have here the result in two families, where the residues are colored according to the ones with which they co-occur. So there's a bunch of clusters of co-mutated residues we could find. Sometimes they really make clusters, like this red one here, but most of the time they are a little bit scattered all across the structure. You have, for instance, this green one has commutated with representative a little bit everywhere around the structure. So that could be consistent with compensatory effects. And more interestingly, we could find that the mutations that have contribute to robustness of the structure, so that have these negative values of structural deviation, they tend always to be the ones with high probability of co-occurring with others. So these are the ones here. So the mutations that confer robustness to the structures frequently are co-selected with all the others. The question then was how these interesting mutations can contribute to the overall robustness of the fold. And so the answer here is frustration. So, well, this is not my feeling when scratching my head around the question, but this is a natural property of amino acids in protein, which describes the energetic conflicts within folded proteins. So this is quite frequent property occurrence in nature. It's found in about 10 to 15% of internal interactions in all proteins. And so that decreases the stability of the overall folds, but increases the ability to create function through binding with other proteins and also evolvability. So by looking at this property in our effector families, we found that the mutations that increase robustness tend to vary a lot in this frustration property. So these are the ones you have in green here. By looking into more details then at the phylogenetic trees in effector families, we found there was a correlation between the variation in frustration and the structural divergence of these effectors. And more precisely, we see that a decrease in frustration, as you see here, is often correlated with more drastic change in the effector structure. So that provides a model through which these effector families can remain stable over long evolutionary time, but also may evolve new functions by keeping, even if they keep this conserved fold. So to summarize these insights into the evolution of structurally conserved effectors, I showed you that the effector repertoires of fungi is organized across well-conserved structural families, and just a few of them. But we have ancient folds that persisted in spite of sequence and potentially functional diversification. And that the repatterning of frustration at the surface of these proteins can explain part of the robustness and evolvability of this structure. So we have basically these two scenarios where the ancestral effector could either evolve towards more frustration and remaining then keeping the same structure, or change a little bit in structure and releasing frustration. So this you can imagine cycles of these evolutionary trends that maintain these families stable over time. All right, so that's what I wanted to tell you about. I'd like to thank especially Marc DJAMEI, my collaborator from Curtin University, with whom we had this very fun project. My group here for all the other nice work they do. I would like to invite the French speakers in the audience to check this book for a very broad primer on plant interactions with their environment, including pathogens. And finally, I thank you for your attention, and I will be happy to answer questions if time allows.
Thank you, Sylvain. So are there questions from the audience? Yes, one.
Hi. So I guess you would keep these structural families, you could keep them for two reasons. One is you have a scaffold and you evolve new functions, or one, you keep similar functions. I mean, did you look at, for the ones that you know functions, did you look into them? Do they keep the same functions or likely evolve something totally different?
Okay, well, so I cannot answer definitely to that because, well, as far as I know, there's no cases where we have known functions for several effectors in the same family and very well described. Yeah, so both scenarios are definitely possible, and I would bet on different functions, even within the same family. So the overall fold being more like a platform for evolving this exposed residues and changing interactions with host molecules. But yeah, that would be very interesting to do, and that's something I would like to work on in the future.
Maybe just to follow up on that question, so in one of your slides, you have exemplified one family, which is KP4, which is known to be toxic to fungi.
Yes, yes, I see which one you mean. Yeah, true. So this one has fold, which is analogous to ustilagotoxin, right? And that is used in competition between strains.
Could it be that some effectors are also toxic to fungi and not for necessarily involved in plant
It's possible, so that's connected to, for instance, the work of Bart THOMMA, who's has found that some of these effectors actually influence the microbiota instead of the host directly. Yeah.
Okay. So I could then take one or two questions from the audience. So then one is a little more technical, but it's maybe a concern for many people here is what is the AlphaFold accuracy in prediction?
So we have in our data sets just 5 effectors for which the structure has been solved by crystals. And in those cases, the analogy is very good. So then the next thing I would say is that so we took all the possible care we could with the analysis by so using a threshold to remove the poorly predicted structures based on the AlphaFold metric. But also using this deep HMM approach, we had an independent validation approach in a way.
Okay.
Sylvain, very nice talk. I have a question concerning this conservation of the structures, even if the sequences are so divergent. I mean, if we assume for a plant immune system, if you have basically to recognize few structures and you trigger cell responses against the pathogen, you would have the universal weapon. Wouldn't you expect that this is unlikely to evolve this way? Well, so again, I don't have the answer really, but so there is actually one example of this SCFE1, which is an effector from Sclerotinia, which is recognized by what was first discovered because it's recognized by an RIP from Arabidopsis. And so this belongs to a family which is conserved across several fungi. And the funny thing is, so the overall structure is required for recognition in Arabidopsis, but just parts of the protein are sufficient for recognition in other plants, such as Nicotiana, for instance. So in some cases, the plants are able to recognize the overall folds, but not always. So it's still a mystery why, how it works and why this evolved this way.
Well, thank you very much. We still have to keep in time. We have some little time, so we're still okay. Thanks very much, Sylvain, for your talk. Then I will call Paul BIRCH. Then he's coming from University of Dundee, UK, and he will speak of cytoplasmic effectors and endocytosis, so a second example. Thank you, Paul.
So thanks very much to the organisers, especially to Barbara, who couldn't be here, which is a real shame, for inviting me, a new mycete person, to come and talk to you in a plant-fungal interaction session. So this really is a very, very big room, isn't it? As somebody this morning said that she felt a little bit like Beyonce. So I think I could do an impression. If I could sing, I could do an impression of Phil Collins, but I think you'll be okay on that front. So yeah, my talk's going to be very similar in some ways. It's going to make some connections to what Nick was talking about earlier on. And we're looking at Phytophthora infestans, which is a cause of potato and tomato late blight. Is this work? Yes, good. As it spreads through the tissue as mycelia, it forms these structures called haustoria. We'll just home in on one over here. It's quite an intimate interaction with the plant cell. The plant cell wall's removed. This is a major site of secretion. We get apoplastic effectors, we get enzymes, cell wall degrading enzymes secreted here, but we also get cytoplasmic effectors delivered inside the plant cell where they can modify proteins, processes in the plant to create susceptibility. And we are very, very much interested, like Ellie and Nick and so on, in finding out more about the secretion pathways, but also the delivery and the uptake of these proteins inside the plant cell. The proteins we're interested in are RXLR effectors because of the conserved amino acid motif that's present, usually 30 to 40 amino acids downstream of the signal peptide. So to help with us studying proteins which are delivered inside the plant cell, we focused on those effectors that we know go to very discrete locations when they're inside the plant cell. So this particular effector up on the top left of your screen, O4314, we already know what it does and we know where it goes to. So it goes inside the nucleus in the plant cell and in the nucleolus as well. So this was cloned with its signal peptide and a C-terminal MRFP fusion in a Phytophthora expression construct and expressed, transformed into Phytophthora expressed along with free GFP. So we grow these transformants in vitro. We can see that the GFP, there's a histone antibody been used here, just to show that you're detecting proteins inside the mycelium, but not usually in the culture filtrate unless they've been secreted there, like O4314 has. If we take this into the infection, we can see that the secretion is taking place actually at the haustorium. So if you draw a fluorescent intensity plot across the, let's use this one, across the haustorium, then you can see the MRFP signals outside, on either side of where the GFP signal is. To look a bit further at infection, we have some transgenic plants that we're looking at here, transgenic Nicotiana benthamiana, which is a host of Phytophthora. That is expressing cyan fluorescent protein, CFP, fused to histone H2B. This is in the nucleoplasm, but it's not in the nucleolus. So we look at an infected cell where we can see the haustorium. We can see, can you see it? Yes, good. There's RFP within the nucleus and especially within the nucleolus. So we draw our intensity plot across the nucleolus. We can see as the blue fluorescence goes down, the red fluorescence goes up. And if you go to a neighboring uninfected cell, you don't see that. So as I said, we're interested in the whole journey from the initial translation of these proteins right the way through to them reaching their destination inside the plant cell. We have evidence, has been published by Barbara and Nick previously, that a lot of these RXLR effectors are going through unconventional secretion pathways. So we're still trying to work out exactly what's happening there. We're also interested in their journey between the cells, and there's actually a talk on Thursday morning by Susan Breen from our lab. And she's going to be talking about extracellular vesicles. And we see that a lot of RXLR effectors are either within or associated with EVs. But what I'm going to be talking about is Heischer's work, and she was looking particularly at uptake inside the plant cell. And she focused on clathrin-mediated endocytosis. So our starting point was fortuitous because Silke Rabacek's group had already published a hairpin construct for silencing the Nicotiana benthamiana clathrin heavy chain family members. So we took that and had a go with it, and sure enough, it silences CHC. We then looked at endocytosis by expressing the pattern recognition receptor FLS2 fused to GFP inside the benth cells. And we can see that that's normally at the plasma membrane, unless we treat these plants with the ligand, the PAMP, bacterial PAMP FLAG22, which then causes endocytosis within the first half an hour to an hour after treatment. If we silence the clathrin in these cells, then there's a significant reduction in that endocytosis. We also noticed that if you co-express FLS2 with a plant RAB5 GTPase, ARA6, and do the same experiment, treat with FLAG22, then you see that they're going to exactly the same puncti inside the plant cell. So we then silenced ARA6 using a hairpin construct, and sure enough, what we saw was a reduction in endocytosis of FLS2. So where do these proteins localize during infection? We can see that if we express in clathrin light chain in this case, fused to GFP, it localizes round the outside of the haustorium, probably in association with the extrahaustorial membrane. ARA6 also does the same thing. But if we silence these two proteins, interestingly, what we see is a considerable reduction in Phytophthora infection. So again, that was something that Nick was showing earlier. So that means that Phytophthora actually wants and needs these proteins to be present. It wants the components of clathrin-mediated endocytosis. And so what we're seeing over here in terms of the localization around the haustoria, the result of that is actually of a net benefit to the pathogen. So we look at the delivery of these effectors then inside the plant cell to their destination. O4314 will accumulate in the host nucleolus, and it's much easier to see over here on this fluorescence trace. As the blue fluorescence drops, the red fluorescence increases. That isn't the case where we silence the clathrin. It also isn't the case where we silence the ARA6. So it seems that both of these proteins are in some way important for the effector to get inside the plant cell, reach its destination. So silencing clathrin can have various pleiotropic effects. So the next step for us was to avoid the silencing, move away from that, and actually follow a biochemical route to try and capture plant endosomes. So for that, what we did was we used an already published method. We adopted that and then adapted it slightly. And it was basically a series of sequential ultracentrifugation steps that allowed us to enrich for endosomes. So after the first two spins, where we're removing debris, large organelles such as nuclei and so on, there's then a 1 hour spin of 30,000 G. That will capture endosomes in recent plant studies. If we take the supernatant and then spin that for a further 100,000 G for an hour, then we take the pellet there as well. That can capture the rest of the endosomes, if you like, and usually the smaller ones. Okay, so we have 4 samples, the initial total input sample, the 2 pellet samples, and the final supernatant. And if we do this in plants which are expressing either CLC, the clathrin light chain, or the ARA6, we can see that there's an enrichment at the P30 and P100 samples in both cases. If you look at this cytoplasmic marker, UGPase, that tends to be reduced in where we have these spins. It's much more readily available within the final supernatant. And if we look down a confocal microscope at what's resuspended from these pellets, we can see lots of small green vesicle-like shapes of around about the right size for medium to late endosomes, medium-sized endosomes. If we treat these samples with the detergent Triton, then we lose these samples. So it suggests that they are indeed membranous. And that can be confirmed here. So ARA6 GFP vesicles captured during the spins are also stained by FM464. The thing I'll draw your attention to here is there's a lot more FM464 staining than there is just where we see the ARA6 GFP. So that's something to bear in mind here in the next step, which is for us to then take the method for trying to enrich the endosomes into a Phytophthora infection. So we've got infections in A and B with a Phytophthora expressing the RXLR effector O4314 fused to RFP. In C and D, we've infected with Phytophthora expressing an apoplastic effector, SCR74. We enrich for clathrin or for ARA6. In the case of the first two samples, we also get an enrichment specifically of the O4314, which we can't detect actually in our input samples. As far as the SCR74 is concerned, we can see it throughout. There's probably more of it there in the supernatant, but we can see it throughout all of those samples. However, if we use a load of other plant membrane markers, so we have TOX75 from the chloroplast membrane, it's also enriched. We've got ARF1 from Golgi, also enriched. We've also got calnexin from the ER, that's also enriched. So, just going back there, that might help to explain that we're seeing a lot of membrane vesicles here, many of which are probably the result of damage to the plant cell as well. So that prompted us to try a next step, which was to perform an IP, to pull out either ARA6 GFP or CLC2 GFP from the 30,000, 100,000 spins. So that's what we did. So at the top here we have infection with Phytophthora expressing the RXLR, O4314, or the apoplastic effector SCR74. We've got clathrin that's been enriched by the 30,000, 100,000 spins. We can see enrichment of ARF1 and the calnexin here as well. We can also see that the O4314 is there. And the same is the case over here for the infection with SCR74. But when we pull down the CLC2, we do not co-immunoprecipitate the ARF1, the calnexin. And we don't actually co-IP the SCR74, but we do co-IP O4314 strongly in the 30,000 spin case. That's exactly the same if we're expressing ARA6 GFP in these plant cells. Again, the only thing that's co-IP'd is the O4314 from the 30,000 spin. So that really encouraged us to take another step, which was to perform a proteomic study, an unbiased approach, if you like, using non-transgenic Phytophthora infecting Benthamiana. So we have Benthamiana that's either uninfected or it's massively infected. This is spot inoculations of spores on the leaf surface. In both cases, these plants are expressing our ARA6 GFP. We're then going through our process of trying to enrich the endosomes and then an IP as well of the ARA6 GFP out of the 30,000, 100,000 spins. And all of these, including the input, are going in triplicate into mass spec from different replicates. So what do we see on the plant side? So these two volcano plots at the top, they show in red the proteins which are enriched from the ARA6 GFP IPs from either the 30,000 or the 100,000 spins. The large blobs are actually ARA6 itself, and what we have in these two cases is a total of around about 600 proteins, 200 of which are in common between the two sets. Amongst these, happily, are 80 proteins which are to do with vesicle trafficking. So that gave us a great deal of comfort that we were indeed capturing endosomes. So what do we see on the pathogen side? So looking in red over here, we detect apoplastic effectors, but only in the total input samples. We don't detect them after the enrichment of the endosomes and the final IPs. In the case of the RXLR effectors, we can detect some in the total and in basically in all of our samples. But some of the effectors are only found in the IPs of ARA6 from the 30,000 or from the 100,000 spins or from both. So it looks like we have indeed captured endosomes and captured with them a number of RXLR effectors that we couldn't detect in the total input. So that suggests to us, with the same sort of caution that Nick was mentioning, that we have evidence of clathrin-mediated endocytosis for the uptake of these RXLRs into the plant cell. There's a number of next steps for us. We want to know more about how the endocytosis is being triggered, what the form is that the effectors are in when they enter the plant cell, critically, how they would be released from endosomes. Nick was mentioning rupture of those endosomes. That may well happen. We can see proteins that within our data set that would be cytotoxic, cytolytic proteins. There could be membrane fusion events as well, with the outer plant membrane at the endosome. Or it could go through retrograde trafficking and be released through something like the INRAE system. So all of that's to work out in the future. So this has been quite a team effort behind all of this, but a special thanks to Heisha, who's certainly done most of the work. Okay, thank you.
Thank you very much, Paul. So any questions from the audience? So I see Ralph moving.
I'm moving slowly. I can't help myself. Great talk. So, like the Magnaporthe story, do you see any sort of special structure that could be involved, or there's nothing suggestive of a structural feature?
You mean in comparison with the BIC?
Say to the BIC, for example.
So I think we do, actually, and it's just something that Barbara pointed out to us when we were first looking at this about 15 years ago. The first confocal images we had were, whenever we look at an Arixolar effector when it's secreted, it's part of a sort of near a neck band at the base of the haustorium. If we look at that in more detail, we can see that it's made up of membrane from both the plant and the pathogen. So there's something quite interesting going on there, and that seems to be where we see the Arixolar effectors.
Okay, and do you think the actual motif has any role in being associated with these vesicles in any way?
We do, but again, we're still at something of a hand-waving stage, but we can see that the Arixolar motif is a cleavage motif.
Yes.
It's required for association with extracellular vesicles for us, which suggests it's going down a different secretion pathway. So there must be some sorting mechanism. It's not the same as the one Nick was referring to, or we don't have evidence of that, but we have something that might be doing a similar job.
Okay, thanks.
Any other questions? Okay, so I have one from the app. So how effectors which are into the endocytosis system are not degraded by the vacuolar system, because maybe this vesicle then could be directed to the vacuolar plant system and be degraded?
Yeah, we don't have the answers. I mean, I think if there was some sort of rupture, you get the effector release very quickly after entry. So that could happen in the two ways I was mentioning. Before you actually get a mature multivesicular body or whatever going to the vacuole. A number of effectors are also ones which are blocking the proteasome. So there's a lot of things to take into account. It's going to be complicated to work out. Yeah.
Okay. So the last question from the app is, when these endosome structures, do they move within the plant cell? Would you see, if you see kinetics of movement of this vesicle with the...
Do you know, Marc-Henri, I've not seen it myself.
Okay.
But the people in the lab might have done.
You need a movie.
I think need Petra to come and answer that one, I think.
Okay.
So, yeah, they'll be moving.
So maybe I have a very last question. So just because the recent paper released on the sorting of the effectors could be involve codon usage in Magnaporthe, do you have any clues of that in oomycete?
Again, we don't, we haven't looked, but I think we're seeing something that may be very, very analogous to the system within malaria. So they have effectors with a very similar motif to the Arixolar. That is cleaved co-translationally, actually. So as it's translated into the ER. So there's something happening there. There's then post-translational modifications. So you get N-acetylation, and that's been reported for Arixolars as well.
So post-translational control could be involved.
Yeah. So that seems to be the route to follow for us at the moment.
Thank you very much.
Okay.
So no more questions. Thank you, Paul.
The next speaker is Tim Friesen from the USDA in Fargo, and he will be talking about the diverse functions of Parastagonospora nodorum effectors.
Okay, first, thanks to the organizers for a chance to speak in this session. So in our lab, we work on the necrotrophic pathogen Parastagonospora nodorum on wheat. So it's a little bit a different pathogen. It actually induces programmed cell death to complete its life cycle. So this slide here is about 20 years of work. On the left, you see all the necrotrophic effectors that we've identified and characterized. On the right, all of the host genes. In each case, on the left, the necrotrophic effectors are all small secreted hypothetical proteins. And on the right, we have susceptibility genes or sensitivity genes. So when we infiltrate a leaf we infiltrate a leaf with the necrotrophic effector that has the host gene target, you end up with necrosis. And then that is also used in the pathogen when it's infecting that same leaf. So I want to focus here on the TOX5 SNN5 interaction. This is the most recent effector that we've cloned and characterized. So quite some time ago, we identified TOX5 in culture filtrates and mapped the sensitivity gene, which we called SNN5, corresponding to TOX5, on chromosome 4BL in the wheat genome. That sensitivity, as you can see here, is mapped as a single gene trait in red. Then when we inoculate with the pathogen that secretes the TOX5, you can see that the yellow line here is the association with disease. So the pathogen is using TOX5 to target SNN5 to cause necrosis and to cause disease, and then ultimately to complete its life cycle. So our goal at that point was to clone the TOX5 gene and then characterize the gene and the protein. So we started with a GWAS approach. We collected 197 isolates from all the different wheat growing regions of the United States, which includes winter wheats, spring wheats, durum wheats. So we had a fair amount of diversity. We then inoculated these 197 isolates on this line, LP29. LP29 is the differential line. It only contains SNN5. So we inoculated on that line, and we identified the strong marker trait association here, indicated by the three green spots. And you can see down here the three markers either land in the gene or just adjacent to the gene, which shows the power of GWAS in fungi. So from there, we looked at that gene. We started by knocking the gene out. So you can see here at the top, we have SN2000. That's the wild type isolate that has TOX5. We have an ectopic transformant here, which you can see both of these have nice disease. When we knock this gene out, we lose that ability to produce necrosis. And if we look at that across the segregating host population, you can see that the two isolates that have TOX5 give us this strong association with SNN5, but the knockout, and also here we have SN79 1087, which is a completely avirulent isolate. Both of those just flatline on this, so there's no association with SNN5. But then when we put TOX5 back into 79 1087 or put it into the avirulent isolate 79 1087, we gain that association with SNN5. So this is pretty good validation that this 6735 is indeed TOX5. So the next thing we wanted to look at is what is TOX5 doing, and potentially what is TOX5 doing in addition to its induction of programmed cell death. We know that when we infiltrate it, we get programmed cell death, but we also have some evidence that the other necrotrophic effectors like TOX1 and TOX3 have secondary functions or potentially even primary functions, alternate functions, other than just the induction of PCD. So here we have a 12-hour time point using confocal microscopy, and you can see on the left here in each one of these, the left is the wild type that has the TOX5, and it's inoculated on LP29, our differential line that has SNN5. And over here we have the knockout. And you can see that the wild type quickly penetrates the epidermis. Over here we just have almost a searching for a penetration point. At 24 hours, similar, we continue to have this mycelial growth on top of the surface, whereas in the wild type, you have colonization of the epidermis, more extensive colonization. By 48 hours, you can see we're starting to get colonization here, but on the wild type, we have a strong colonization of both the epidermis and the mesophyll. But notice at this point, we still don't have any necrosis. So this is one of the things that we're learning is that the necrotrophic pathogens are actually growing out in front of the necrosis, and the necrosis happens after it's built its biomass, rather than the effectors being put out in front and it coming in behind and living on the dying tissue. So by 72 hours, you can see this is we're starting to see some necrosis. You can see here by the red, the cellular breakdown in the top of the mesophyll, but the pathogen is building its biomass out in front. Over here, we only have just a small amount of colonization of the epidermis. And you can see here the mesophyll area is completely clear. Similarly, at the 96-hour time point, we just have colonization here of the epidermis, nothing in the mesophyll, and you can see that here. The mesophyll is clear, but in the wild type SN2000, you can see strong cellular breakdown, and it just gets worse by 120 hours, where you have complete tissue collapse here. But the pathogen has built enough biomass to then be able to sporulate or complete its life cycle out of this. Whereas in the knockout, the pathogen never makes it into the mesophyll. It just stays between the epidermal cells, and that's as far as it colonizes. So one of our questions was, does TOX5 have a secondary function similar to, say, TOX1 and TOX3, and then maybe it acquired the PCD function later. So here we looked at the TOX5 with SNN5 without TOX5. So we flipped this around to also eliminate the necrosis, the ability to produce necrosis, but we put TOX5, the wild type, back in, but we knocked out SNN5 in the host line. So when we did this, you can see that the pathogen colonizes the mesophyll. We still don't get necrosis, but we get a strong colonization of the mesophyll, indicating that TOX5 may be facilitating that colonization even in the absence of SNN5. So the next thing that we really wanted to know, or we've wanted to know for a while, is really what makes a necrotrophic specialist a necrotroph. Okay, so why can it function in this environment? Because things like the oxidative burst and the upregulation of defense response genes, PR proteins, chitinases, all those kinds of things can't be a benefit to the pathogen, but the pathogen has evolved a mechanism to use programmed cell death, to use the defense response to complete its life cycle. So Guyan Kariwasam in our lab, he's a postdoc, has done a deep RNA-seq, enough where we can get enough of the pathogen. So he presented on the host side yesterday, if you were at that talk. So I'll just talk about the pathogen side here today. So we just collected leaves at the same time points that I just showed you, and then performed the RNA sequencing and looked at both host and pathogen. So I just have here at the top and down the side the different time points, so you can kind of see what's happening at each one of these at each one of the points in the infection process. So this is the necrosis-inducing effectors, which includes tox5, tox-A, which is one of the first ones that was cloned, and then tox1, which also has a secondary function. And you can see all of these, the peak production of these is all at 24 hours. But the necrosis doesn't actually happen. We don't see the first necrosis until 48 hours later. So the pathogen, well, we'll move on to the other classes. So we know that wheat produces a lot of chitinases. So we looked at chitin binding effectors. So each one of these, if it has a green mark, it was validated by qPCR, but each one of these has a differential expression from the 0 time point. So you can see these, I'm just highlighting the peak expression, but you have peak expression here long before the effector is producing, or the peak expression of the necrotrophic effector. So this is likely protecting early on, allowing the pathogen to build biomass before it goes into that necrotrophic phase. We have induced defenses, so these are polysaccharide deacetylases. So you have 3 of these. Those are likely involved in reducing the chitin to chitosan to make it less visible by the pathogen, or I'm sorry, by the host. Those are also happening throughout the early stages, and then this particular one is late. The oxidative burst is another thing that the pathogen has to navigate, and so this. So here we have 3 thyroidoxins, which are involved in neutralizing reactive oxygen, and also the superoxide dismutase. These are involved later on here in the 96-hour time point, which is where we really have a lot of reactive oxygen. But we also have some that are involved in the early phases here. Lastly, we have cell wall degrading enzymes, which if you look at the old literature on necrotrophs, you would think that these cell wall degrading enzymes would be used in penetration and early getting into the cell and colonizing. But we see, so we just have 4 here: cutinase, cellulase, xylanase, and pectinase. And you can see there's 35 of these that are differentially regulated. But most of these are involved quite late in the response, which means they may be involved in more nutrient acquisition than they are in actually penetrating of the host. So I just want to end with this slide. So this is just a direct comparison of kind of 2 extremes of. So on the left here we have the necrotrophic pathogen that we're working with. On the right we have a very good pathogen, stem rust of wheat. But I just want to highlight the differences between the 2 interactions. Here, necrosis leads to susceptibility, whereas in the stem rust and many biotrophs, necrosis leads to resistance, where this pathogen here has evolved a mechanism to keep the host alive, and that's how it sporulates, whereas the necrotrophic pathogen, in this case, it has evolved completely differently in that it's found a way to kill the cell and to complete its life cycle. So with that, I'd like to thank the people that actually did the work. And so my lab, as well as some former lab members and several longtime collaborators. I'll just advertise quickly here for the 3 things that we have left from our lab, 2 posters, and then Ashley will be giving a talk on Friday. And then, as I'm the editor-in-chief for MPMI, I have to give a plug for MPMI. We have a effector, since this is an effector section. We have effectors at the interface of plant microbe interactions, special issue coming up. If you have any questions about that, or if you have something you'd like to submit, just let me know. That, if there's time, I'll take questions.
Thank you very much, Tim. So I have several questions from the app. And then the first one is whether you would consider that Parastagonospora is an hemibiotrope.
Okay, I get that question all the time. So depending on how you define hemibiotrope, I would define hemibiotrope as something who has a biotrophic feeding structure. So in that case, no. It would be more like Zymoseptoria. But yes, if you just define a hemibiotrope as something that has a latent period, yeah, then almost everything is a hemibiotrope.
Yeah, so there's more questions from the app, and then is whether could TOX5 be involved in PTI suppression in early colonization before inducing the cell death?
Yes, that's possible. We don't have any evidence of that, but yeah, that's possible. Yeah.
So there's another one, whether the person that asked questions would be interested to know your thoughts on the evolution of the tox effectors. Do you think that the necrosis was originally the primary function, and were the secondary functions first and then the necrosis functions evolved?
Yeah, once again, that's another question that we get a lot, and it would just be a complete guess as to what came first. But I can see that what we call the secondary function of these was initially the primary function, and they evolved a sort of a recognition process. I can see that order being logical, but once again, we don't have any proof for that.
Mm-hmm. So I still have questions. So if SN, so Parastagonospora nodorum, is actually growing ahead of the necrotrophic zone, as it is a pure necrotrophic. So I think it means cell to cell in the mesophyll.
I'm not sure I understand the question.
Whether is it actually growing ahead of the necrotrophic zone?
Okay, so yes, it is growing ahead of the necrosis. So it's building biomass in front of, and it's about a 48-hour delay from the necrosis to the advancing part of the...
I think it means after the necrosis is induced to other cells. So is that happening?
Oh, that I'm not sure about.
Mm-hmm. So I think we should move on. Yeah, so thank you very much, Tim. Very nice talk. So the next speaker is Armin DJAMEI, and he's from the University of Bonn, and he will talk about the lessons to learn from a gall-inducing fungus.
Thank you very much. Thanks to the organizers for inviting me. Do I have to click somewhere, or is it opening on its own?
You have to escape.
Push escape.
It's there, but it's not there. Oh, no, it's there. All fine. Thank you. Yeah, I would like to share with you a journey of roughly 10 years where we did different type of screens on a biotrophic pathogen, and we are working basically on Ustilagomedes. It's a smut fungus which causes these dramatic galls on all aerial parts of the plant. And I got fascinated by this pathogen because it's a very intimate relation. If you compare, if you look at this confocal picture, looking at the red fungus here labeled and the green plasma membrane of the host. And this interaction is governed basically by secreted effectors. And so we study these effectors since now 10 years, as I said, in a systematic approach where we do localization screens, we look at the interaction partners, and we do functional assays and try to overlap these results. And I would like to pick out 3 different screens today and show you what we learned from this and what we learned by overlaying these results, to make an educated guess about effector functions, but also to learn something about the plant immune system. So the first screen which we performed, or one of the first screens which we performed still at the Gregor Mendel Institute, was done by basically asking a very principal question, which is an obvious one. Every microbe which is landing on a plant is getting recognized by pattern recognition receptors, and this is triggering the pump-triggered immunity responses. And if one wants to be successful on the plant, one needs to suppress these initial recognition responses, either by avoiding recognition or by suppressing the signaling in order to survive. And so we just simply screened roughly 300 candidate effectors of Eustelagomedes. It sounds simple, it was not so simple, and this was done, work by a very talented former PhD student of mine, Fernando Navarrete. And he basically systematically went through all these effector candidates, expressed them in Nicotiana bentamiana, took leaf discs, and then by luminol-based assay, challenging the plants with chitin or with flagellin, he was triggering a ROS burst, which looks then like this. It's a luminol-based assay. And the higher the curve, the more ROS burst occurred. So red would be, in this case, here the big curve here. I don't know if you see the mouse. No. You can, basically the red curve is the cherry expressing control plant, and the other lines would be then expression of an effector. So suppressing this ROS burst response of the plant. And so the surprising thing was, we did it in a heterologous system, that testing roughly 291 effectors, we found 34 effectors which suppressed pump-triggered immunity responses on the ROS burst level in this heterologous system. And that was a nice starting point. And when we mapped then these effectors in the Eustelagomedes genome, we saw that 10 of them were basically mapping on the same locus in a cluster. And that was surprising to us. So he tested chitin as well as flagellin, and you see here basically the area under the curve plotted, and the smaller the bar is, the more suppressive in terms of ROS burst suppression these effectors are. And he analyzed then 2 of them more in detail, but I will not summarize this because it's already, or I will summarize it because it's all published. But the key point which I would like to make here is that basically the clustering of these effectors is not only because they are linked to biotrophy. In this case, the cluster was really focusing on a specific aspect which needs to be covered, suppression of PTI. Although the sequences of these effectors are divergent. And when we tested specific functions for them, we could also show that they are not targeting all the same protein, but different parts in this cascade of the signaling. Another screen which I would like to point out was basically targeting the question, how does Eustelagomedes causes these dramatic galls? How is this growth controlled or misregulated? And one key hormone signaling pathway in plants for growth is obviously auxin signaling. Just to remind you here, you see the 3 components which are conserved across all land plants, which are found in auxin signaling. It's an activating ARF, auxin response factors, which are transcription factors. Then you have the AUXIA proteins, which are repressors binding to the ARF, and they are recruiting a co-repressor, which is topless, or from the topless family members. And this complex is basically the off status in the auxin signaling. And when auxin is accumulating inside the plant cell, then the AUXIAs get targetedly degraded, and this is then de-repressing the auxin responsive genes, because then the ARFs can go active. So having this in mind, we took a reporter. We took DR5, it's a reporter for auxin signaling. We co-expressed them, and this was a screen done by a former postdoc of mine, Janos Bindic, and later followed up by Mamouna Khan. And this screen basically was revealing, we looked at the reporter expression upon co-expression of an effector, was revealing a number of effectors inducing auxin signaling in the plant when we expressed single effectors in Nicotiana bentamiana. And again, when we plotted them on the genome, again, there were 6 effectors which were all auxin signaling inducing, and they were clustered again in one cluster found, although sequence-wise they were not all related. So that was, again, I hope you see the overlap here, that we have specific functions covered, and they are in one cluster then combined, although maybe they are interacting in different levels inside this cascade. And when Janos was looking for the interaction partners by MassSpec and QIP, and then also later by Yeast2Hybrid, he identified topless to be the interaction partner of 5 of these effectors which were inside this cluster of the tip effector cluster. So that was super interesting. But what was also astonishing, when we took all the auxin signaling inducing effectors which are binding to topless, and we did a screen against all 300, and we found in total 10 of them, we saw that all of them are within this cluster, within the group of 34 effectors suppressing rust burst. And that gave us a new hint that in plants, obviously, topless has somehow a role with rust burst. And we basically showed this then genetically in different publications, that topless, like proteins, are regulators, positive regulators of the pump-triggered immunity response. And this is a conserved thing here. We have shown this in Nicotiana benthamiana, in maize, as well as in Arabidopsis. So our current model for the role of tip effectors targeting topless is the following. We have under the off-state, non-infected plant, we have topless suppressing JA-ethylene signaling pathways, suppressing auxin signaling, suppressing BR signaling pathways, anthocyanin biosynthesis, and it's acting as a positive regulator of the pump-triggered rust burst. And then when the pathogen is coming in, it's secreting tip effectors, in total 10 known ones so far, and they are suppressing topless functions, and thereby they are de-repressing the JA-ethylene signaling pathway, the auxin signaling pathway, growth is initiated, BR signaling goes up, and anthocyanin biosynthesis is induced. And as the hormone signaling pathways have also a lot of antagonisms within the plant, we observe a downregulation of SA signaling. So this is all fitting in that way if you look at the transcriptome of maize infected by Ustilagomades. Still, of course, we couldn't solve at that time, or we are still on it, to answer the question, how can we induce a gall? How is Ustilagomades able to induce galls? And gall formation is basically a process which is found in many plant pathogen, plant pest interactions. And galls are basically plant-formed organs which are induced, formed by the activity, most likely of effectors or pathogens in whatever way. So they are induced organs which are basically pathways from the plant which are redirected in the purpose of the pathogen. So that is for the effector field in Ustilago, obviously the holy grail question: is there a single effector necessary? Do we need many of them? Do we need other cues additionally? Do we need cell type specific expression, or doesn't matter? So there are many questions here to answer in order to answer how do we get this kind of new organ formed in the plant upon infection. And to answer this question, we went back to the principles and asked, okay, what is one of the principal necessities to induce a gall? If you look at this surface of a maize leaf here by raster electron micrograph, you see a nice pattern of pavement cells. And if you then look at the galls, it's obvious this is also structured, but the cells look different. There had been cell division happening, there has been cell expansion happening. Also here in this cross-section, you can nicely see this through a chalcofluor stained gall in the plant. You see that there are elongated cells, cell expansion happened. You see a lot of cell division, both in the epidermis, in the mesophyll cells, as well as in the bundle sheet cells, so basically across the whole leaf. And so we decided to go for a screen to find effectors which can induce either cell expansion, cell division, or both. And for this, we went for stable lines. This was a real group effort in the starting, still at the GMI time, and then going through the IPK time till I landed in Bonn. And we had generated roughly 300 transgenic Arabidopsis lines expressing one by one Arabidopsis effectors, Ustilago effectors, sorry, and we call them the Arafactor lines. And then a screen was performed looking at control plants versus effector overexpressing plants, and we got a lot of different interesting phenotypes which can be followed up. But one line was popping out which was very interesting to us, which was discovered by Mamouna Khan, and she found this line here, which is somehow resembling us to what we looked for plants which are having an asymmetric growth, irregular cell divisions possibly. And so if you look at the raster electron micrograph here from this control hypocortical, you see again nice lines of aligned cells. And in comparison, the overexpression of IAG1 induces cell division as well as cell expansion, as you can see here in these irregular shapes. On the epidermis, if you look at the leaf epidermis, you see again here a nice mosaic of pavement cells. And would be the IgG1 overexpressor showing piles of cells coming out, getting different forms. And basically, it's obvious a very strong growth phenotype. So as we talk about effector candidates, we had, of course, to confirm now also that this IgG1 is really an effector, not just some intrinsic protein which is not really secreted. So we looked at certain features which are typical for effectors. We looked at transcriptional regulation. IgG1 is, as you can see here on this transcriptional regulation, upregulated during 3 to 4 days post-infection. So it's the time where galls are formed. If you look at this reporter strain of Ustilagomades expressing M. cherry under the IgG1 promoter, you can see here in magenta basically the hyphae, which is a cutted leaf here. And you can see that you find the hyphae in the bundle sheath cells expressing the effector, but also in the epidermis and also in the mesophyll cells. So it's expressed not cell type specifically, at least from our reporter. Maybe we need a reporter with a higher turnover. And additionally, we tested, of course, secretion, and using the signal peptide of IgG1, we can demonstrate secretion by the hyphae here, showing the signal around the hyphae. So then we went on for the interaction partners, and we did this in collaboration by a co-IP mass spec approach with our friend Alada Zanter. He is sitting in Hungary, and he identified a conserved kinase, which I cannot yet disclose to you, but it's a family, a serine threonine kinase, and this kinase has orthologs and maize, and we cloned them, tested them, and they are interacting nicely in yeast to hybrid, but also in a split venous approach, we can nicely see that they are interacting both in the cytosol as well as in the nucleus with IgG1. Mamuna then went on and tested the effector for certain domains which might be binding to this kinase, and she identified that the very C-terminal part of the effector is specifically binding to this conserved kinase. And if you co-express this kinase, or first single express it, you see it's mainly nuclear, the signal. But if you co-express it together with IgG1, we observe a redistribution inside the cell towards the cytosol, as well as you still see signal in the nucleus. And so our current model is the following. We have a conserved kinase in plants which is nuclear in the off state, and when the infection happens, IgG1 is expressed, it's translocated into the plant nucleus, and then it's pulling out the kinase to the cytosol, and this cytosolic localization of the kinase leads then to cell expansion and cell division, which we observe here. And with this, I'm at the end. I would like, of course, to first thank the people which have done the work, the whole lab, the former members of the lab, my collaboration partners which were helping me over the time, and the funding agencies. And I would like to thank you for your attention, and I'm open for your questions. Thanks.
Thank you. I mean, is there any questions from the audience? Yep.
I've got a question for you. Can the fungi secrete the phytohormone, like the auxin or cytokinin to stimulate the growth?
Yes. So Ustilagomades is producing auxin on its own. It's fungal derived. It has the TAM1, TAM2 biosynthesis pathway for auxins, and it has been deleted, this pathway in Ustilago, but it has no gall phenotype or no impact on virulence. And this is understandable if you consider that there are many other effectors which cause auxin signaling downstream of the auxin perception. Yeah? So it's producing auxins, it's producing effectors which induce auxin signaling. It has obviously additional effectors which are not inducing auxin signaling but still target the cell cycle and cell expansion. So it has a whole suite of effectors which target in this direction. Yes.
So there's a question from the app, and then is it whether physical clustering of effectors is a specific of Ustilagomades, or is also conserved in other smut fungi?
Mm-hmm. So to my knowledge, there are clusters also found in other smut fungi, in related smut fungi. The size, I mean, there is synteny found with Sporisorium, there are papers, but also in other smuts you find clusters of effectors. Not all effectors are clustered, but I think I calculated something like 30%, 25% of the effector candidates are in clusters, and the rest of the unknown secreted proteins, which are biotrophy induced, are not clustered.
So,
Yeah, great talk. I'm always very interested in topless. I mean, it's a central regulator of growth and development and has many roles in plant pathogen interactions. If I recall, topless is a central regulator of the JA responsive pathway. If that's true, but you were suggesting that it perhaps is regulating auxin pathways. Is it through its negative regulation on jasmonic acid pathway? Is that how you think that topless is working?
I mean, topless is binding both. It's a negative regulator of the JA signaling,
Right.
as well as of the auxin signaling, and we observe induction of both pathways, but our reporter with which we were screening was targeting the auxin part. But in my model, I showed that basically it's also inducing the JA signaling pathway if you overexpress the effector. Okay. And we have found one effector which was very interesting, maybe to add this. It's also published, it's JC1, it's work from Darino et al., also a former talented postdoc of mine. He has shown that JC1 is binding to the C-terminus of topless, and it's mainly targeting the ethylene JA signaling pathway, whereas the other tip effectors which interact with topless bind to the N-terminus, to the TPD domain, and interfere with the ear binding motif of topless. So the effectors target the same protein family, but there's still specificity within.
Thanks for a great talk. This is perhaps a naive question or too soon for this, but have you tested whether adding a nuclear export signal to your conserved kinase in a mutant background would cause a similar phenotype?
Yes. It's it's fresh work, yeah? So it's unpublished. We are having these lines created. We will look at the phenotypes to test, of course, if this is holding true, that the activity needs this export-import thing. But so far, I cannot answer it yet. Yeah.
Hello, I mean, very nice talk. So I was wondering about this last story that you were telling us about the effector that is phosphorylated by this kinase. So have you done any in vitro assay to identify any serine or threonine residue that is being phosphorylated in the effector, and if you know any motif that might be like involved in the function of this?
I didn't say that it's phosphorylated by the kinase. I just said it binds to the kinase and it brings it out of the cytosol. So we have no yet indications that it's getting phosphorylated itself. So it's basically, so far we assume it's taking it out of the nucleus and thereby redirecting the signaling cascade to the outside, to the cytosol, and then we observe the phenotype which we observe. But this still needs to be tested with further experiments, obviously.
Okay. Thank you.
So thank you very much. I mean,
thank you.
So the last speaker of the session is Bart THOMMA from the University of Cologne, and he will talk about plant pathogens manipulation of the host microbiota. Sorry.
Okay. Thanks, Andrea, for the introduction, and thanks to the organizers for inviting me to be able to present some of our data here. And I would like to first start off by showing you a video of our favorite pathogen, and that favorite pathogen is called Verticillium dahliae. And you see two seedlings, two tomato seedlings, in fact. This pathogen is not a pathogen of tomato only. It infects hundreds of plant species, but we use tomato very often as a model. And by now, you slowly start to see that these two seedlings develop differently. The one on the left-hand side keeps growing, gets bigger, looks actually quite happy, but the one on the right-hand side already by now looks quite pathetic. And that's because it's infected by this verticillium fungus. That's actually a soil-borne fungus. Infects the roots of plants and then grows into the xylem vessels where it starts to sporulate, it starts to spread. And that's why, basically why the sap stream in the xylem gets blocked. And the consequence is very clear. In this case, its plant is very much stunted. It doesn't develop well. It shows necrosis and it may eventually die. what I would like to do at this moment too is at least give you a definition of what I think effectors are. I mean, we obviously are in an effector session. I mean, many of the speakers before me have talked about effectors, but still I would like to stress that my definition at this moment, and I hope by the end of the talk you will be convinced of my definition. My definition at this moment is that we should see effectors as molecules that are secreted by microbial pathogens, by plant pathogens during attack to enable host colonization. And you see that there is a bit of text that I sort of made vague, wiped out. I realize that many of us, and also myself, we considered effectors as molecules that target host physiology, and maybe many of them actually targeting host immunity, to suppress host immunity, to manipulate host physiology in order to enable host colonization. But again, I hope by the end of the talk you understand why I think this part of the definition is maybe not so important anymore. And to explain you a little bit of why I think this is the case, I'm going to tell you a story that starts over a decade ago with an effector that we cloned from Verticillium, and it's actually, at that time, it was the only effector of Verticillium that was known to be recognized by an immune receptor in tomato, and that immune receptor was called VE1, and the effector that is recognized by this immune receptor we called AV1 for mediating avirulence on VE1 plants. And in the bottom pictures, you see tomato plants, and you actually see the effect of deletion of this effector from the gene, or this effector gene, I should say, from the genome of Verticillium, but now not on plants, tomato plants that carry the immune receptor, but plants that lack the immune receptor. And I think if you compare the middle and the right plant, the wild-type Verticillium that infects tomato versus the AV1 deletion strain that infects tomato, then it's clear that the deletion strain causes less symptoms. The plant looks bigger. It's not healthy. It's not as big as the mock-inoculated plant, but clearly it's less diseased. So it's an important virulence factor, we thought. And we spent a lot of time trying to find out what does this effector do. And we failed basically to find a function for this effector, and at one moment we realized actually there's, maybe in all the assays that we did, there are some hidden messages that tell us that we're on the wrong track. And so the first message is that whereas we typically think that effectors are strictly implanta-induced and are not induced outside the host plant, for AV1 we found that actually it's sort of constitutively produced. It's always produced. Yes, it's produced obviously in the plant. That's where it's recognized. But it's also produced in vitro. Now in vitro is not so relevant obviously for the biology of Verticillium. But then if we grow Verticillium in soil or in soil extracts, then we also see expression. The second hint, I think, came from the fact that we tried to produce AV1 in heterologous systems, in microbial systems, in order to get protein for biochemical assays. And also there we were very much unsuccessful because whatever we tried, we never got soluble protein. And so we realized that AV1 in E. coli, also in yeast, actually is misfolded, it's brought into inclusion bodies in E. coli, and that's where that's then where the protein is deposited. And this is basically where we started to realize that maybe we were on the wrong track. We were thinking about functions in suppression of host immunity, but maybe we have to think about functions outside the host, for instance, in the soil where AV1 is expressed. And take the problems of expressing this protein in E. coli as a suggestion that perhaps this is an antimicrobial protein. And so we started testing this. We isolated AV1 from inclusion bodies. We refolded it carefully. We tested whether the refolded protein stimulates an immune response in resistant tomato plants, which it did. And then we started using this protein for functional assays. And so the first thing we obviously did was we started screening microbes for sensitivity to AV1. And the panel here shows with the green line incubation in the presence of AV1, and the red line is a water control, no AV1 is present, that this particular bacterium, Bacillus in this case, is sensitive to this AV1 product. Now, Bacillus was not the only bacterium that's sensitive, but we used that one as a model. And in the bottom, you see some electron microscopy pictures where it's clear that treatment with AV1 leads to lysis of bacterial cells. So then we reasoned, okay, but now we would like to test whether this is relevant in planta. So we went back to this wild-type verticillium and the AV1 deletion strain, and we performed inoculations in plants. We did this in tomato, we did this in cotton, and then started to profile the microbiota of these plants. And we didn't find enormous shifts in the microbiota, nothing that I would call a dysbiosis. But when we started zooming in, we did see that particular taxa of bacteria were repressed in the presence of AV1. And when we focused into particular taxa that were repressed by AV1, we started to realize that actually these were antagonists of verticillium growth. Antagonists for verticillium growth in vitro, but also when we treat tomato plants with these particular bacterial antagonists and then perform infection assays of the wild-type verticillium strain and the deletion strain, we clearly saw an effect of these bacteria on verticillium colonization, and the moment verticillium does not produce AV1, the impact is even bigger. So with that, we had the first evidence, or at least the first suggestion, of an effector protein that maybe targets the host microbiota in order to help verticillium to colonize the plant. And this is where we realize that this actually totally makes sense, because we also know, first of all, that plants are able to recruit microbiota and to recruit beneficial microbiota that help them to withstand stress and that also help them to withstand pathogen infection. And actually, on top of this, we know that in the soil, and especially the rhizosphere, is a very microbe-rich compartment where there's a lot of competition going on. So all of this made sense, but then obviously the question for us was, isn't this now a very strange effector that evolved in this interaction, or are there actually many more of these effectors? And I will not go into detail because most of this, actually all of this, what's mentioned here on the slide is published. By now we identified a handful of factors that have antimicrobial activity, all from this verticillium fungus. And what we take from it is many of them have expression patterns in different stages of the life cycle. So some, like AV1, seem to be expressed constitutively, more or less, throughout the life cycle, but others are expressed only in soil or only at very late stages of the verticillium infection. They all have selective antimicrobial activity. There's no broad spectrum antimicrobials, and then some are antibacterial and others are antifungal. The one additional effector that I'm going to spend a little bit of time on is the second avirulence molecule that we know from verticillium. So after cloning the first one, a new resistance source has been identified in tomato. Again, here we cloned the corresponding avirulence molecule a couple of years ago. Also this one, like AV1, despite the fact that there is no sequence homology at all, also this one seems to be expressed in soil. Also this one, when we produce it after refolding procedures, we find that there is antimicrobial activity, but now antifungal activity mainly. This gene seems to make a contribution to virulence, not as strong as AV1 does, but it clearly does. But now it depends on soil type we test. So we tested various soil types. On some soils, in tomato infections, we see a contribution to virulence, but in other ones not. And here, in addition, we did some tests to see whether it may also contribute to the ability of verticillium to colonize soil, so in the absence of the host plant. And we clearly see that in extracts from unsterilized soil where we have a full microbiota, the deletion of this AV2 gene makes that verticillium is less able to colonize, whereas in sterilized soil this difference is not observed, clearly showing that it's the microbiota that is targeted by this AV2 effector protein. So I would almost say, without too much effort, we find many of these effector proteins in the genome, encoded in the genome of verticillium, that display antimicrobial activity. And this, I honestly have to admit, also surprised us a lot. And it got us thinking, and it also made us think, where do these effectors actually come from? And the hypothesis that we came up with is that perhaps this antimicrobial activity of these effector proteins is something that's very ancient, that was already encoded in ancestor fungi that colonized land before land plants arrived, when fungi mainly had to compete with other microbes for carbon in terrestrial systems. And could it be that perhaps these effectors later were co-opted in all the extant fungi that we know nowadays that colonize as saprophytes, or that cause disease, that colonize plants or animals, and in those contexts either still kept this antimicrobial activity or evolved other functions. Now, if this is true, then we should find distribution of such effectors much more widely. And so what we did here is simply we look at all kinds of fungal genomes, pathogens, non-pathogens, saprophytes, and we predicted effector catalogs and asked the question, are there now effectors that are basically conserved throughout all these fungi? And there's actually quite some that we can find like this. And if we now go to the left-hand side of the plot where we have these effectors that are the most widely distributed, and we start picking candidates here and test them, then actually we again relatively easily find antimicrobial activity. So one of these candidates is very widespread and also present in verticillium. So this candidate we produced again, we tested, and yes, it indeed again has antimicrobial activity, now a combination of inhibition of bacteria and a number of yeasts. But if this hypothesis is really true, then of course we should also be able to find these type of activities outside verticillium. And I'm going to show you two examples. This is an example from the white rot pathogen, in this case of avocado trees, Rosalenia necratrix. We try to predict effectors with potential antimicrobial activity that are expressed during host colonization. We found effectors like this, produced them, and two of the candidates that we tested indeed display antimicrobial activity. Again, in this case, a combination of bacteria and fungi that are inhibited. And then we said, well, but can we take this a step further? Can we go beyond plant pathogens? So what we actually did is look in the genome of a lichen fungus. So now we look at a symbiont, a mutualist, a fungus that associates with algae to generate lichens. Also there in the genome, we can predict effectors. We can predict effectors with potential antimicrobial activity that are clearly also expressed during interaction within this lichen, so in the interaction with algae, and also here, some of these candidates, when we produce them, we can clearly show that they have antimicrobial activity. So by now, we're really convinced that the production of these types of effectors among fungi and in fungal genomes is very much widespread. Now, this is the last slide I'm going to show you. One of the things we're trying to do now is, given the fact that we are now convinced that these types of activities are so widespread, we think it would be very good to have a tool that helps us to identify those effectors with antimicrobial activities. And I have to admit, at this moment, we mainly do this based on domains that we recognize, based on homologies that we see. What we're trying to do at this moment is use AI, use machine learning to come up with a tool for effector proteins, so typically a bit larger than the typical antimicrobial protein predictors that are around nowadays. So we have composed sets of known antimicrobials in the size range of typical effectors, and control sets that do not have such activity, that are equally sized, and that come from similar type of organisms. We predicted structures of all those molecules and try to extract all kinds of structural features from these effectors and use that to try and set up a machine learning approach to predict antimicrobial activity. And the graph in the bottom shows, although there's, at this moment we have not done any tweaking, we're still very much early on in this process, but it seems like the prediction works really well because in fungal genomes we already end up with an accurate prediction rate of about 86% without doing any tweaking. So we're very much convinced that this will help us to make a tool to be able to recognize antimicrobial activities among effector proteins. So with that, I've come to the end of my presentation, and basically I hope that you're convinced with me that at least not all effectors target host immunity, but they can also have other functions besides targeting host immunity and host physiology in order to stimulate host colonization. And maybe you also have a little bit of faith in our hypothesis that it could be that this is actually an ancient trait that was there in the ancestor of many effector proteins that we recognize as important for pathogenicity nowadays. And with this, I'm going to stop. These are the acknowledgements. This is a picture of most of the people in my group at the University of Cologne. The people highlighted in bold are the ones that contributed to the story I told you about today. And I specifically would like to mention Nick Snelders, a former PhD student in my group, who basically was the PhD student who initiated all this work on looking at antimicrobial activities among effector proteins. And with this, I'm going to stop and thank you for your attention.
Thank you, Bart. Are there any questions from the audience?
Hi, Bart.
Hello.
Maybe I missed this, but so is AV1 and the other effectors that you mentioned, for that matter, so they're delivered into plant cells as well?
No, we don't think so. No, these are all what we think to be apoplastic effectors. Yeah, we don't have any evidence for these effectors to be deposited inside host cells. We know of effectors of verticillium that go inside host cells, and they have the typical modulation of immunity or modulation of host physiology activity. But all these effectors that we think have antimicrobial activity, they are what we tend to think apoplastic effectors. Yes.
So there's a question from Megan McDonald, and she's asking whether you could comment on the host range of the antimicrobial activity of the effectors.
So, like I already mentioned, they all have very narrow spectrum activities, so there's not very broad activities, especially the ones that have antibacterial activity. There we see that the spectrum of bacteria is very limited. In general, they tend to be antagonists, but for instance, if we look at AV1, we know it acts against bacillus, and then we know, for instance, it acts against fingomonad bacteria, but not against pseudomonas. I mean, the spectrum of bacteria that's inhibited is very narrow, and that goes actually for all of these effectors. So, I mean, we basically either determine these spectra based on microbiota profiling in planta, which obviously is quite laborious to do, or testing of culture collections, individual bacteria. But then we never see that many bacteria are inhibited. It's always a very specific group. Mm-hmm. So we have one that targets sphingomonads. There is another one that targets actinobacteria. We think we see very little overlap. So it seems to be highly specific molecules that pick out particular taxa in a plant.
So I hear what you're saying, that they have antimicrobial activity. But oftentimes when you're working in the laboratory, you're working with semi-sterile soils. And it's very clear that AVR-V1 is a very strong effector. If you lose it, even on susceptible plants, right? So I think there's more to the story than just interfering with the microbiome in the soil. What about in planta, possibly?
No, so for AV1 itself, we have evidence that it does so in the soil as well as in planta. Okay. Actually, the profiling we did in planta, because in soil it's much more complex. Right. But I do agree with you that AV1 has a very strong virulence effect, yes. And we actually believe that the antimicrobial activity is not the only effect.
Thank you.
There is more to this. But again, this comes back to this hypothesis. We get more and more convinced that the antimicrobial activity is a sort of ancestral activity, ancestral function, but it acquired other functions as well. And for these other functions, we don't have evidence that they contribute to virulence yet, but I'm also not convinced that we can explain the full virulence effect only through the microbiota.
Okay, I agree with that. Good.
Yes, next question.
Yes. How do you explain apoplasmic effectors and virus infection which occurs in the cytoplasm?
How do I explain apoplastic effectors and, sorry?
And virus infection, which is known to occur in the cytoplasm. So how do you relate? apoplasma and virus infection?
Well, so I think for these, I mean, when I talk about these effectors, I'm obviously mainly talk about fungal effectors, and maybe we can extend this a bit to effectors of bacteria or of oomycetes. I mean, that remains to be seen. I think viral infections, of course, is a completely different story, and I don't know immediately, I don't immediately see, let's put it like this, an enormous contribution of the microbiota in the interaction of a virus to a plant. Okay. I think that, at least for sort of that my thinking is a little bit that fungi in this total microbiota have to fight for their place, and in this niche in the plant, they have to fight with other microbes to colonize this niche. This becomes even more relevant the moment nutrients are released, which may attract other microbes as well. And these, I think, are processes that may not be so extremely relevant in a viral infection. So I think a viral infection, when you think about viral effectors, I do not want to suggest that microbiota manipulation is a major function to be expected for such effectors.
I'm afraid we don't have more time for questions, but there are many in the app, so I invite you and the other speakers to please check the app and reply to them. So thank you very much, Bart.
Thank you. And just for the speakers, so you can answer anytime today to the question asked on the app, please do, because tomorrow they will be away.